So I would like to show you just a small example of how you could use GeneVestigator for finding genes involved in C4 photosynthesis. And for those who are new, GeneVestigator is a software for analysis of gene expression developed at Nebion. Of course, before starting using any software, you need data to analyze. And for that, you need to do experiments. So thanks, Andrea, for doing this, and other co-authors, of course. So you saw those pictures already. So here, the authors compared two species. One was C4, Chinandropsis chinandra, and the other one was C3, Hasleriana. And those species are closely related. So, and they harvested tissues, including those leaf gradients, from a very small, hardly visible leaf to that big, green, mature leaf. And the important thing, as I understand here, is that photosynthesis doesn't take place in that small leaf. It's not ready for that yet, but then it comes, either C4 or C3, depending on the species. And they also harvested other tissues like roots, stems, seeds, and so on. And they obtained gene expression data by RNA sequencing technology. So we thought it's a good experiment to include to GeneVestigator. We did that. So if you open GeneVestigator, you see something like that. Here are the experiments related to the 324 project, three of them. In that case, the reads were mapped to Arabidopsis transcriptome, and the pipeline was established at Nebion mainly by Jean-Marie, who is sitting there, so thanks. And here you have various tools that you can use for analysis of those experiments. So what I decided to do for the examples, I used differential expression tool. So in that tool, if you open it, oops, sorry, you can, no, I'm wrong. So if you open it, you can choose an experiment that you want to analyze. So I chose the one I just showed. And then you can define which samples you want to compare. So I thought I will compare leaves of the C4 plant, those that can do photosynthesis, so that's those that are able to run the C4 cycle, versus everything else, so versus all other samples of the C4 species and versus all the C3 samples. So I wanted to know what is specific for the C4 photosynthesis. No. That's this one. And so that's the result obtained. So each dot here represents a gene. So those genes that are red are expressed more in Chinandra C4 photosynthetic leaves, and the green dots are expressed less in Chinandra photosynthetic leaves versus all other samples. So, and then I thought, okay, then I take just one gene and look at it in a different tool, in GeneVestigator, where you can, where you can now look at each sample and look what happens with the gene in each sample. So that's the gene. So that's now the result from the samples tool. So here you have each sample of that experiment. All those belong to the C3 plant and all those to the C4 plant. And a dot is here now an expression value. So if it's more here, then it's low expression. If it's more at the right side, then it's becoming highly expressed. And now those gray samples belong to the leaf gradients, C3 and C4. And what you can see here is that in the C4 plant, when the plant grows, the gene becomes more and more expressed. So it correlates, so the gene expression correlates with the ability of the leaf to do the photosynthesis. However, in the C3 plant, when the leaf grows, gene expression doesn't change. So that's a good correlation. And also, if you look at the C4 plant, non-photosynthetic tissues don't really express that gene much. And then the good thing in that tool is that you can have many experiments at the same time. So you could have here a lot samples and look at the same time what happens with the expression of that gene. So here we have another two species, Prostrata C3 and Rosea C4. And also here, the C4 species express that gene highly, whereas C3 species, in C3 species, the gene expression is low. And that were mature leaves. So that corresponds basically to this. So I thought, okay, that gene looks like C4 photosynthesis related gene. And then I, of course, looked what it is. And so you are, of course, laughing. It's very known gene, found partially, I think, by you. And it encodes a pyruvate transporter from the cytosol to the chloroplast in the C4 cycle. That's Wikipedia, so don't blame me. So, of course, now with that, there's probably no real scientific value, but for us, it was a good indication that actually the pipeline that we established at Nebion actually works, and we can find C4 photosynthesis genes. And now it's actually up to you if you would like to do something more. So even in that one comparison, there were more genes, and I think one of those spots was this GNAT transcription factor somewhere. So maybe one could compare that list to Andreas' list or something like that. And of course, you can always open GeneVestigator and define your own comparisons, whatever you like. And we are constantly adding new experiments. So there is a good chance maybe to find something more interesting.